Hello, this lecture covers hereditary environmental influences on childbearing and conception and fetal development. So in this lecture, we're going to cover conception, fetal development, and genetics. Genetics. Well, genetics is the study of particular gene. It provides the tools to determine the hereditary components of many diseases, improves our ability to predict susceptibility, onset, progression, and response to treatment. Currently, there has been a shift from genetics to genomics. Genomics refer to the study of the entire genome. And so genetics impact everyone. So that is why it is very important for nurses to understand what genetics is all about. Essential competencies for all nurses is to know or to have expertise in genetics. Nurses are responsible for collection of data, so we need to collect family history using standardized symbols and terminology. It is also important to develop plans of care that incorporates genetic and genomic assessment information when we are dealing with our patients. We want to also facilitate referrals for specialized services if there is any genetic disorder. And it's very important to assess clients' knowledge, their perception and responses to genetic and genomic information. And also with genetics, there has been advanced testing. So with improvement in diagnostic capabilities, that has actually led to a lot of good benefits. It has resulted in earlier diagnosis and enabled individuals who previously would have died in childhood to survive into adulthood. And so thanks to technological advancement, some disorders appear more often within ethnic groups. For instance, Tay-Sachs is very prevalent among the Ashkenazi Jewish. And this Tay-Sachs is a nervous system disease or disorder. So it's very prevalent among the Ashkenazi Jewish. Cystic fibrosis, on the other hand, is more common among Caucasians. And then the sickle cell is prevalent among Black Americans or African Americans. Let's take a look at some chromosomal abnormalities. Chromosomal abnormalities, this is a major cause of reproductive loss, congenital problems, and gynecologic disorders. And then we have the sex chromosome abnormalities. And so these sex chromosome abnormalities, there are two major ones. We have Turner syndrome, which is also known as monosomy X, and Klinefelter syndrome, trisomy XXY. So these are two sex chromosomal abnormalities. Let's look at Turner syndrome first. Well, this Turner syndrome is a disorder or chromosomal abnormality in which females are affected. So these females who are affected by Turner syndrome are missing all or part of an X chromosome. And so usually they have amenorrhea or infertility. They also experience delayed puberty, heart defects, or certain learning disabilities, and they appear short in height. They do have short height. So that is what this Turner syndrome is all about, is sex chromosomal abnormalities that affect only females. So only females are affected by this Turner syndrome. On the other hand, Klinefelter syndrome affects males. Klinefelter syndrome is caused by the addition of an X chromosome. Normally, males have 46 chromosomes with one X and one Y. However, in Klinefelter syndrome, there is two X chromosomes. And so males who are affected, they have low testosterone, reduced muscle mass. They also have body and facial hair. They produce little or no sperm. And so this is a congenital condition which happens before or at birth. So these are congenital problems, Turner syndrome or Klinefelters. So once again, Turner syndrome affects females and Klinefelters affect males. More chromosomal abnormalities. This one is called the autosomal abnormalities or autosome. So these are other type of abnormalities other than the sex chromosome. These are abnormalities of chromosome number, and we have a good example of Down syndrome. Well, Down syndrome is a genetic disorder caused when abnormal cell division results in extra genetic material from chromosome 21. So that is why it's called trisomy 21. So when you take a look at this image, you look at this baby in the slide on the slide, you would no doubt notice that there is a low ear set. So low ear set is usually one of the remarkable signs of Down syndrome. Simian crease is also a sign of Down syndrome. So you see the simian crease where you have a transverse line running down on the palms. This is what simian crease is, and that is an abnormal finding. It's usually seen in monkeys. So when you have a baby who has a simian crease, that's not a normal finding. So low ear set and simian crease, those are signs of Down syndrome. And of course, you see that protrusion of the tongue. And also with these kids who have Down syndrome, they have developmental delays coupled with intellectual disability. Abnormalities of chromosome structure. There is also translocation, which is caused by rearrangement of parts of the chromosomes. It can also be deletion, and this is due to missing parts of chromosomes. So in deletions, you have missing parts of the chromosomes. And in inversion abnormality, you have a rearrangement in which a segment of the chromosome is reversed end to end. So that's what happens in inversion or type of abnormality of chromosome structure. Patterns of genetic transmission. Well, patterns of this genetic transmission can be unifactorial or multifactorial. Under unifactorial, this is when a single gene is controlling a trait, disorder, or defect, such as in autosomal dominant, autosomal recessive, and linked chromosomal abnormalities. Now, these are the multifactorial patterns of genetic transmission. And so the multifactorial is the most common genetic malfunction. And with multifactorial pattern, there is a combination of environmental and genetic patterns. For instance, cleft lip and cleft palate. A cleft palate is an opening or a split in the roof of the mouth that occurs due to lack of the tissue due to lack of fusion of the tissue during development in the womb. So you may see a child who is delivered and the child has a cleft palate. So that is a pattern of genetic transmission, which is multifactorial. And also you could have a baby that is delivered with cleft lip, and that is due to inability of the lip to form properly in utero. So those are examples of multifactorial patterns of genetic transmission. Neural tube defect is also another example, and congenital heart disease falls under this pattern as well. Teratogens. Well, teratogens are agents in the fetal environment that either cause or increase the likelihood of a birth defect. So this is why it is very important for women who are pregnant to avoid any of these things on the slide, because these are things that could increase the likelihood of any birth defects when a woman is pregnant. Radiation, alcohol, drugs, hormones, cigarettes, German measles, lead, mercury, all those things can cross the placenta and invariably have a teratogenic effect to the developing fetus. So the key is prevention. So you want to prevent exposure. Key is to prevent exposure to any of these teratogenic agents. So you don't allow a woman who is pregnant to have a chest x-ray or to go through radiation. So she cannot do that because that will be harmful to the fetus. And this is why we encourage patients who are pregnant not to consume alcohol because that can have a teratogenic effect to the fetus. Drugs should not be used. Only class A drugs or medications should be used, and to be sure they need to talk to their doctors before they use any drugs. Not even over the counter, they have to check with their doctors. Although acetaminophen is safe, but you always want to tell them to check with their doctors before they use any medication. And of course, a cigarette is a no-no because it will have some effect on the fetus. In general, class X medications are harmful. Medications such as methotrexate can have a teratogenic effect to the fetus. Only class A is safe during pregnancy. The ovum. Well, the ovum is a mature female reproductive cell which can divide to give rise to an embryo after fertilization. And so this oogenesis is the process that produces the female gamete known as the ovum. So oogenesis is that process that produces ovum, and ovum is a mature female reproductive cell which can divide to give rise to an embryo, usually after fertilization. Spermatogenesis, on the other hand, is a process that produces the male gamete called sperm. And it takes about, with each single ejaculation, look at how much sperm that is released, about 35 to 200 million with each ejaculation. And that's pretty amazing. So now we're going to turn our attention to fertilization. Well, fertilization equals conception. And so this is the fusion of haploid gametes, the egg and sperm, to form the diploid zygote. So the sperm fuses with an ovum to form a new diploid cell or zygote. This fertilization is a brief period of time to unite. So after ovulation, the ova remains fertile for about 12 to 24 hours post-ovulation. The sperm is also active for about 48 to 72 hours. Usually within the first 24 hours after having sex, that's usually the time that could take place, especially when you have a couple that is very fertile. So that fertilization would take place, and it has to be during the time of ovulation. So a woman can only conceive during ovulation. If you are having sex and it's not during ovulation, it will not happen. So conception or fertilization can only take place during the time of ovulation, and this fertilization takes place in the outer two-thirds of the fallopian tube known as the ampulla. So fertilization takes place in the ampulla, and this ampulla is the ampulla of the fallopian tube, which is the outer two-thirds of the fallopian tube. That is where fertilization takes place, and it will only happen during ovulation when the cervical mucus is thin and elastic. So that is the only time a woman can conceive. Now, after fertilization, there will be rapid mitosis, which is cell division. And usually this is 3 days after fertilization. You have what is called the formation of the blastocyst. So the blastocyst gets formed, and then this blastocyst implants itself into the endometrium and surrounds itself in maternal tissue. This implantation usually occurs about 7 to 10 days after fertilization. So this is the process of this mitosis. So this is 12 to 15 hours after having intercourse or after fertilization. Then you have the division into 2-cell stage. It goes to 4-cell stage, becomes 8-cell stage. Then it becomes a morula, and then this morula now turns into a blastocyst, which is usually noted around day 3, and then it continues. Embryonic period. Well, the embryonic period is the beginning of 3rd week through the 8th week after conception. So that is what we use to describe the fetus or the product of conception beginning at week 3 through the end of week 8. So you have that formation of the zygote. The zygote now becomes an embryo. So it is an embryo until the end of week 8. From week 9, it becomes a fetus. So during this embryonic period, there are basic structures that needs to develop. So basic structures of all major body organs are completed during the embryonic period. And this is why it is very important for a woman to abstain from any of those teratogenic agents during the embryonic period. During this embryonic period, the tissues differentiate into essential organs, and this is most vulnerable to teratogens. The embryonic period is the most vulnerable period for teratogenic effects. So if a woman is exposed to any of those agents that we mentioned, radiation, lead, alcohol, measles, all those things, if it is exposed, if the woman is exposed to them during this embryonic period, it'll definitely harm the fetus or have a teratogenic effect on the fetus. This is what the embryo looked like around week 4. So at week 4, so see all those major organs have developed. You see the yolk sac, the placenta, umbilical cord, the heart, the eye, brain, all those things are already, there. So all this fetus, fetus will now do as the weeks continue is to develop. At week 6, the week 6, these are the things that is noted during the week 6. There is head, the head rapidly developing over the chest. Heart is now 4 chambers. Facial development occurs. Nasal, you have the eyes, the ears, all those things are now, appearing. So once the end of 8 weeks comes, once the week 8 ends, you now have the fetal period, and this fetal period begins from week 9 after conception. So it starts from week 9 after conception and ends with birth. So it is a fetus until this baby makes his or her grand appearance. Now you have a neonate or a newborn. So don't forget, remember what the embryonic period is and the fetal period. This is your fetus around week 9 to 12. The fetal heartbeat may be detected by Doppler, usually around 10 to 12 weeks. And this is a picture of a Doppler. That is what it looks like. Week 13 to 16, the fetus grows in length. And this is when quickening may be perceived by moms, especially between 16 to 20 weeks. And remember, we said quickening is that fluttering sensation. So you feel like something is moving inside your abdomen, and that's your baby. So that quickening begins around 16 to 20 weeks. Around 17 to 20 weeks, the skin is covered with vernis caseosa. And the vernis caseosa is it's like a cheese-like substance. So it's like look like this baby is poured with cheese, like somebody poured cheese all over the baby's body. And that is the vernis caseosa. It actually protects the skin of the baby. And we have lanugo. This lanugo is fine down in hair. The lanugo development starts around 17 to 20 weeks. And you have these fat or brown fat deposits. And these brown fat deposits contain small mitochondria. So it's like the powerhouse of the baby or the fetus. And the primary function is to generate body heat in animals or newborns that do not shiver. So this is the purpose of the brown fat deposit in this in fetus. 21 to 24 weeks, the long alveoli begins to form. Remember, we said that the lowest age of viability is 21 to 24 weeks. So because of that lack of surfactant, that is why if you deliver a 21-weeker or 22-weeker, the chances of survival may be very slim, because this is the period that the long alveoli is just beginning to form. A surfactant production begins in lungs, and fetal respiratory movement is noted around this period. And so with surfactant, it reduces the surface tension throughout the lungs. So that is why surfactant is very, very important. 25 to 28 weeks, now the chances of survival increases. The chance of survival increases with immediate and prolonged intensive care. Eyes begin to open and close after 26 weeks. This is what the fetus look like around 29 to 32 weeks. There is presence of toenails, of subcutaneous fat is present, the skin is pigmented. 38 weeks, now it's almost there. You have a well-developed ability to exchange gas. So the fetus has a well-developed ability to exchange gas. Lanugo on shoulders only, as well as the back area. So that lanugo, that fine downing hair that covered the whole skin, is now disappearing. So you will only see lanugo on shoulders and back only. The vernix will only be seen around major body creases. So it's no longer covering the whole body. However, if you have a premature baby, if you delivered a baby prematurely, you will definitely have the presence of vernix and lanugo. But as the fetus makes it to full term, this starts to disappear. So you will only see vernix in major body creases. So at this time, let's talk about placenta. You cannot talk about pregnancy without placenta. Both are interwoven. So now placenta is described or is defined as the interface between mother and fetus. Placenta has several functions, and the functions include gas exchange, metabolic transfer, hormone secretion, and fetal protection. So the key function of the placenta is respiratory and gas exchange by simple diffusion. Placenta plays a huge role during pregnancy. It has metabolic functions in the sense that placenta synthesizes certain substances into glycogen, cholesterol, and fatty acid. Is a transfer organ in the sense that it transfers oxygen, nutrients, waste products, and antibodies. It also has endocrine function, which is with the secretion of certain hormones that are needed during pregnancy. Placental hormones include what is called human chorionic gonadotropin, HCG, and this HCG is the basis for pregnancy test. Remember, when we're looking at the urine test, when we're looking at the blood test, we're looking at the level of the HCG, and if you're pregnant, it definitely rises or increases. So that is one of the placental hormones. Progesterone is the most essential hormone that is responsible for the maintenance of pregnancy. So progesterone is the key hormone that is responsible for the upkeep of pregnancy. It causes endometrium to change into the decidua, thereby providing nourishment for the fetus. Estrogen is also another hormone. Estrogen is responsible for enlargement of breast, uterus, and external genitalia. It enhances uterine activity as the woman's due date approaches. So as the woman is getting ready to have this baby, the estrogen enhances uterine activity. And finally, we have lactogen, or the human placental lactogen. So this promotes nutrition and fetal growth, decreases maternal insulin sensitivity and utilization of glucose, making more glucose available for fetal growth. The umbilical cord, or the umbilical cord, is referred to as the lifeline. So that is how the lifeline between the fetus, the mom, the placenta. So the oxygenation nutrients go to the fetus through the umbilical cord via the placenta from the mom. So that is the lifeline. Umbilical cord should consist of 2 arteries and 1 vein, known as AVA. So remember the mnemonic AVA, A-V-A. An umbilical cord should have 2 arteries and 1 vein. And in some circumstances, you may have some babies who are delivered with only 2 cord vessels, and you usually see that in kids or babies with congenital heart defects. But about 95% of the times, cord vessels should have 2 arteries and 1 vein. So remember AVA, 2 arteries and 1 vein. And of course, there is something called the Wharton's jelly. So you see when you look at this cord, you see that the Wharton's jelly is kind of it is very slimy. So that protects and insulates umbilical cord vessels. So remember AVA. The fetal membranes. Well, at the time of implantation, there is the formation of the amniotic sac. So during this time of implantation, embryonic membranes form. So you have that amniotic sac, and this amniotic sac consists of two slightly adherent membranes known as the amnion and the chorion. The amnion is the inner membrane which contains amniotic fluid. So when a woman breaks her water, when she's ruptured, that is the sac that ruptures. So that is the amnion. The amnion is the inner membrane which contains amniotic fluid. And then you have the chorion. This chorion is the outer membrane, and the chorion has a special features called chorionic villi. The chorionic villi serves as a barrier between maternal blood and fetal blood. Maternal blood and fetal blood never should mix. So maternal blood never mixes with fetal blood. And this is because of that protective barrier that chorionic villi provides. So it has a special feature called chorionic villi, which protects or prevents maternal blood from mixing with fetal blood. So under normal circumstances, maternal blood and fetal blood should never, ever mix during pregnancy. This is a picture of the placenta. So this is the placenta, and this is the sac. So you see that the amnion and the chorion. Amniotic fluid. Amniotic fluid increases in volume throughout the pregnancy. The normal volume range at 40 weeks, it's usually the range is about 700 to 800 milliliters. And so you need sufficient amount of fluid to provide the following functions. So the following are the functions of amniotic fluid. It provides thermoregulations for the fetus. It allows symmetrical growth and development of the fetus, cushions and protects the fetus, and protects umbilical cord. So these are the core functions of amniotic fluid. And so in order for this amniotic fluid to perform these functions, you need to, the woman who is pregnant needs to have sufficient amounts of fluid around the fetus to allow this to function, or to have, fulfill these functions. Volume is important determination of fetal well-being. There is something known as oligohydramnios, and this oligohydramnios is deficiency of amniotic fluid. So when you have a deficiency of amniotic fluid, usually about less than 5 centimeters, and this is usually due to fetal renal anomalies. So if there is a renal problem, that will lead to oligohydramnios. And we have polyhydramnios or hydramnios, and this is excess amount of fluid. So you have a woman who is pregnant and she has too much fluid around the fetus. This is usually seen in about 1% of pregnancies, and is seen in anomalies such as GI problems, Down syndrome, cleft palate, and dwarfism. So you'll see that in those patients whose fetus have those congenital problems. Fetal circulation. This is very important. The blood leaves the placenta via the umbilical vein, and so that blood that leaves the placenta via the umbilical vein is oxygenated and nutrient-rich blood. So it leaves the placenta via the umbilical vein, and then it enters the fetus. Now, after circulating through the fetus, the blood returns to the placenta through umbilical arteries, and the blood that is returning to the placenta via the umbilical arteries is high in carbon dioxide and waste products. So blood supply bypasses the fetal lungs because they are not in use. So in utero, when you and I were in utero, our lungs were not in use. So you could say that the placenta was like the lungs when we were in utero. So the fluid filled fetus lungs. So the lungs were fluid filled, and so the fetus obtains oxygen and nutrients from the mother through the placenta via the umbilical cord. And so that's how the fetal circulation takes place in utero. Multi-fetal pregnancy. This is when you carry in twins. So multi-fetal pregnancy, we have twins. Twins can be monozygotic or dizygotic. If it is monozygotic, it is referred to as identical twins. And if it is dizygotic, they are called fraternal twins. So what is the differences between the two? Well, in a monozygotic twins, you have single fertilized ovum. They usually is same sex, same appearance, though it randomly occurs. There is no association with assisted reproduction in terms of monozygotic twins. So this is a picture of monozygotic twins. So they sharing everything. So they have the same, they shared placenta. They are identical twins. They're a separate amniotic sac, but they shared placenta. And also in this one, so this is a monochorionic diamniotic twins. So in these diamniotic twins, they are sharing separate amniotic sac. However, the placenta is, they have separate amniotic sac, but they are sharing one placenta. You have monochorionic monoamniotic twins. In this type of twins, they have shared placenta and shared amniotic sac, and they are identical. The dizygotic twins or fraternal twins. The dizygotic twins usually they have two separate ova that is fertilized by two separate sperm. So you have two separate ova fertilized by two separate sperm. They have two placentas. So when you look at this, you see that they are not sharing anything. Everybody have their own thing going on. So they have two placentas, two chorions, and two amnions. And they may be same sex or different sex. So in dizygotic, everybody have their own separate thing. They have two separate ova fertilized by two separate sperm, two placentas, two chorions, and two amnions.